Today we will be demoing iBioSim and SPL Designer, two tools that we are going to use to create a genetic toggle switch. SPL Designer is a standalone application as well, but here it is embedded inside of iBioSim. So first we're going to open our project in iBioSim, and we're going to be building the genetic toggle switch using only certain steps, since we are coming from a project that has a lot of parts already pre-built. This is just for time. So first we're going to start off building the TET-R inverter gene. This will open a blank canvas in SPL Designer, and in here we will drop a promoter, a ribosome binding site, a coding sequence, and a terminator. So this is considered with dealing only with the structure of the DNA. So SPL Designer has a nice feature where you can import parts from the SPL stack. This basically allows you to contact a registry online through the internet and grab parts from there. This makes it so that you don't have to type out the sequences yourself or find the part yourself. It's all in a handy registry. So this is all done using the internet, so make sure you have an internet connection if you want to try doing this at home. So we are now specifying every single part in this TET-R inverter gene. And after this, we can save this SPL file and associate it in our SBML model so that we can model this. So here we are creating that model. This is the TET-R inverter model. This is now dealing with SBML, not SPL. So here we are dropping down two species. This represents, the first one is a TET-R, and it represents the input for our model. And the second one is LAC-I, and this represents the output. We are setting the initial concentration to 60 for modeling purposes. And here we are drawing the interactions between these two. Here TET-R, the input represses the promoter, and this promoter we will associate with the SPL that we just built just a second ago, namely the TET-R inverter gene. So after this SPL is associated, you will see the SPL visual glyphs are now dropped into the design. Here is the LAC-I inverter. We have associated the SPL and built this beforehand, but it follows basically the same steps. Now we're going to be looking at genetic toggle, which is the top level hierarchically defined version of this. And here we are dropping down two modules. Each of these modules represents the models that we were just building. One is the TET-R inverter and one is the LAC-I inverter. And here we are going to connect these modules with the rest of the design that we also pre-built. And so the rest of the design basically defines what the simulation is going to do. Here we have the smaller dark green boxes as events with ATC and IPTG going high and low, so that we can demonstrate what happens when this genetic toggle switch is used. So here we click on the analysis view, and we have some parameters that we can set so that we can do a simulation. Right now we have it set to a Monte Carlo simulation. And after you simulate it, which we are doing now, we can click on the TSD graph and see a simulation of what happened. So as you can see from here, the red line is the ATC molecule. GFP, green fluorescent protein, is the green line, and IPTG is the blue line. So in the initial state, GFP is not being produced, but when we introduce IPTG to the system, GFP starts getting produced, and then even after IPTG goes low, GFP remains high. So that's exactly what we want with the functioning genetic toggle. And after a little bit of time, ATC goes high, which causes GFP to go low, and then even after ATC is removed, GFP remains low. So this shows that the genetic toggle has simulated, and it is functional, and our model is correct.